So thanks for coming. Today I'm going to be talking about AlphaFold. Its use, its applications, generally what it is, what's under the hood, how to use it, how to interpret the results, and some of the applications, especially here at the LMB. All right, so why do we need structure prediction? I mean, probably this doesn't need to be said at the LMB, but as you know, first pass, we want to know what our favorite protein looks like, right? So this gives us structural context to biological questions, of course. Some of the common uses are, for example, trying to interpret cryo-EM maps or crystallography, right? So we use structure prediction to be able to fit into problematic regions in our cryo-EM data that can help us interpret it. We can use it to guide protein structure design for biochemistry, for example, figuring out where you want to make truncations and such. You can even use it in molecular replacement and crystallography. And essentially what we want to do is quickly generate hypotheses that we can then test using other techniques, right? And so this goal towards trying to predict the structure of a protein purely from its sequence was assessed by this global experiment called CASP. And so researchers would submit their algorithms, and then they would be assessed against a sequence that they hadn't seen before. And if they performed well, then they would be ranked high in this GDT. So this is a global distance test. And just to show you the history, AlphaFold came out in 2018 with very strong results, and then in 2020 with outstanding results, to a point where now we're considering it in our workflow when we're thinking about different projects. And so here's an example of at the time when their algorithms produced predictions that were very close to the experimental result. Again, these are proteins that they had not seen before. And so there's other similar kind of algorithms that were developed. I'm going to focus mainly on AlphaFold in this talk because it's the most commonly used. It's slightly more accurate, or has better performance on a lot of structures than other things. But I will mention briefly, just going to move this, some of the other algorithms. And when I say AlphaFold, I'm referring to AlphaFold 2, which was the initial one that came out, which was for single chain sequences, and then AlphaFold Multimer came out slightly later, which was for protein-protein and multiple chains where you can actually predict protein-protein interactions. So for this talk, I'm going to use AlphaFold as kind of a collective term here. And what allowed this increase in performance to occur was the development of neural network architectures that can do the predictions. And so to train a neural network model, what was used was existing examples of sequence to structure pairs, right? So we have in the protein data bank a lot of examples of structures where we know what the sequence should be. So if we can train a model to then predict the structure of a sequence it has not seen, that would be very helpful. And one of the key insights that was implemented into the pipelines was the use of a multiple sequence alignment rather than the sequence itself. And so we have our sequence of interest, but if we look at a multiple sequence alignment and look at the sequences that are homologous, we can infer a lot more about the relationship between proteins and the residues in the sequence, right? So here are an example of two residues that seem to be co-evolving. So they're following similar evolutionary paths, and then therefore can be inferred as being close to each other in space. And so things that are not kind of following similar evolutionary paths are probably not going to be close in space. And this is the key insight that was injected into these models that allowed it to then perform really well in the predictions. So when thinking about these structure predictors, you really should be thinking about it's the multiple sequence alignment that's the input. It's important to frame it that way in your mind. And so there's two main classes. There's these multiple sequence alignment structure predictors, which is what I'll call them here. And this is AlphaFold and RosettaFold, which you probably heard of. Also very accurate structure predictor from David Baker, Institute for Protein Design. And then we also, so these are the ones that take a multiple sequence alignment, they generate a multiple sequence alignment, and that gets fed into the neural network. We also have these protein language model structure predictors that were developed. And these are slightly different in that they don't take a multiple sequence alignment as their input, but the evolutionary relationships are embedded within the models themselves, right? So they're a lot faster, might not perform as well, but because they're so fast, they've been applied to these big genomes and allowed a lot of predictions to make it to databases and such. So this is like ESMFold from Meta, which you may have heard of. Again, I'm going to focus on AlphaFold because it's most commonly used, especially at the LMB. And so let's take a look at what's going on with AlphaFold under the hood. we have our sequence of interest. Like I said, the multiple sequence alignment is the input, right? So the first step in this is to do a search against a sequence database. I'm gonna figure this out. Anyway, you can see what I'm talking about. So a search against a sequence database. So there's a few databases that you can look, that you can fetch homologous sequences from to develop a multiple sequence alignment. And then this is fed into a multiple sequence alignment module. So this is the part of the AlphaFold that's trained, right? So the multiple sequence alignment module turns this MSA into, initial XYZ coordinates, depending on these evolutionary relationships that I mentioned earlier. And then it goes into a structure module, which turns that into something that actually looks like a protein, right? Something that has side chains and that actually looks realistic. There's another part of this that is that can be fed into the MSA module, which is actual structural templates. So if we have our sequence, we can find similar sequences in the PDB that should give us insight, right? So this should be an alpha helix if you have something similar to this string of sequences, and that can be fed in. But it turns out this is not as important. And what's really cool is that most of the information that is required comes from the MSA, actually. So usually templates are not helpful, not as helpful. What is very helpful is this recycling step. So at the end of your structure module, you get a structure, and usually it's not very realistic. And so it's fed back in for another round of prediction, and this iterative process results in improvements in the prediction. And so this is one of the also key developments. So you may know, if you've used it, that before it was typical to use 3 recycles, and now it can be upwards of 20 recycles, which we'll talk about later. But essentially, this is an important part of the process. And because the multiple sequence alignment is so important, it's good to talk about exactly what's going on here. So when AlphaFold is fed a sequence, it needs to go look at these databases, right? And so the databases that it uses are UniRef90, Magnify, and BFD. These are, these large databases of sequences from different organisms. And this is usually a rate limiting step, actually, in a lot of implementations because it's slow, right? And so you may have heard of AlphaFold, which is a faster version of this multiple sequence alignment fetching. And it's essentially using a clustered version of these databases and using a more optimized way of pulling out those sequences. And so it's fast. That's the one that we use. I think most people are using a AlphaFold implementation. And so when you hear that, it's the same pipeline. It's just that the multiple sequence alignment comes from an MMSeq search. Yes. So this is very simplified, right? I mean, it's much more complicated, of course, under the hood, but I think as a user, this is probably the level that would be good to understand. And really, in practice, all you need to do is give these things your sequence. And from the sequence, you get your structure. So that's what's really nice about it. And as a first pass, usually it performs quite well. And for AlphaFold, you get 5 structures out. And this is because there are 5 neural networks that were trained slightly differently from each other. And this gives us 5 different chances of obtaining a good prediction, right? It's not always going to be the same one that performs well, but depending on the protein, sometimes model 1 will perform better than model 3, et cetera. And so we get 5 that will be ranked. And the best one usually is the one that's top ranked, but we're going to talk about this a little bit more. What we get also is these multiple sequence alignment metrics. This is very important, of course, because that's the input. And so we're going to talk about what this means. And in terms of interpretation, interpreting your prediction, there's these 2 metrics that are important to understand. The predicted local distance difference test, which is the confidence of every single residue in isolation, and then the predicted aligned error, which is the confidence of residues relative to another residue, right? So let's go through these. Right, so here are our structures. So I'm just going to use an example from a protein that I work on. So this is dynein. Dynein is a microtubule motor. So it moves along microtubules, and it's part of this really large complex with dynactin and this coil adapter. And here I'm just showing one of the subunits of dynein. So this is the light intermediate chain in blue. And it's a nice example because it has this globular domain and then this alpha helices that are connected by these flexible loops. And so let's see what AlphaFold does with this protein. You can see we have our globular domain, and then we have kind of our alpha helices that are floating in space. And it's kind of placed these alpha helices randomly in space, and probably the position of these things are not accurate, right? So it's kind of randomly placed them, but we need a good way to kind of interpret this, right? Typically, you'll get them ranked, and the ranking of these models has kind of changed over the years. with different implementations, what is considered to be the better ranked model than the other ones. It's usually something like average PLDDT, which I'll mention, template modeling score. I find these not very intuitive and not very useful in terms of absolute numbers. And in practice, what you should do really is look at all of the models. And the PAEs usually are more helpful, especially if you're looking at protein-protein interactions. So make sure you look at all of the models if you are trying. The multiple sequence alignment, like I said, is very important because this is the main input to AlphaFold. In this example, we've been able to fetch 3,000 sequences. And these sequences don't cover the full range. Some of them are smaller, right? They're covering different parts of the protein. And there's a term here called MSA depth, which is how many sequences we have. And you can see the MSA depth is low for the NNC termini, and the depth is higher for the middle region of this protein, right? So we're going to get more confident predictions probably of the center of this sequence than we are going to get of the NNC termini. And as a rule of thumb, you want to have greater than 30 sequences, which doesn't sound hard, but for very divergent organisms, it could be actually a limiting step in getting a proper multiple sequence alignment. So it's good to look at this kind of thing. Now, to interpret each prediction that you get, one of the first things, the easiest one to understand, is the predicted local distance difference test. And this is, like I said, a residue by residue score. So it's telling us how confident are we in this, prediction of this amino acid. And we have our 5 different predictions that are overlaid on top of each other here. And we're going along the sequence in the x-axis, and the PLDDT is on the y-axis. And a general rule of thumb here is that if something's above 50%, then it's probably an ordered residue. So it's part of a secondary structure. And if it's below 50%, it's probably going to be disordered. It's a bit hand-wavy, but this is kind of the guidelines that AlphaFold provides. And it generally matches what you see in the models, right? So we have in this prediction that I showed you, we have these loops here that are quite disordered, and those have quite low PLDDTs. So they're probably going to be sitting randomly in space, not necessarily the way it's shown here. And then we have these alpha helices that are probably correct, and they have secondary structure, and we probably can trust them and infer from them, and proceed further with these predictions. The one that's slightly less intuitive is the predicted aligned error. People find this one a bit more confusing. I mean, it is not as intuitive because it's essentially, a residue versus residue plot, right? So we're asking, what is the confidence of one residue versus another? And the logic is a bit strange because what we're asking is, what is the expected error of residue 1 if you align this prediction with the true prediction at residue 2? We don't have a true prediction, a true structure, sorry. We don't have a true structure for this protein. But what's powerful about AlphaFold is that it's able to predict this error just based on the way that the model performs, right? And so if the structure is really true, then if we align it here, these residues should also align here, right? So that's therefore very confident. But if we align here and these two are very different, then probably we're not very confident. So that's what we're seeing here is the error. So the higher this number, the less confident we are. I also see people make this error a lot. So the higher the number, the worse the confidence, right? So you either say we have very low PAE, very confident. So just be careful with your wording with this. And so let's take a look at this. So some very trivial example. Let's say, what's the confidence of residue 198 with itself? Obviously, it's going to be very confident. So our PAE is going to be very low. And everything along this diagonal is a residue with itself. So of course, we have a very strong diagonal. If we look somewhere else, for example, we go up and we ask, what's the confidence of residue 198 versus 113? It's going to be low confidence. So we have high PAE, right? The error is going to be high. So this is a red part of the plot. And obviously, these aren't interesting examples. But what's interesting is if we have subdomains now, right? So we have regions that are close to each other in sequence space that are starting to become confident with a neighbor and the neighbor's neighbor, right? And so these are stretching away from the diagonal. We have these areas with low PAE that look like patches, and these are typical of subdomains, right? If we continue along the sequence, we have this loop that there's not much to say about the PAE plot here. Then we have this other subdomain that shows up. Similarly, it's a patch, right? That's extending from the diagonal. And then we have the subdomains here are interacting with each other. And so these are coming out as patches that are away from the diagonal. So the whole patch itself is kind of sitting off the diagonal. So these are regions that are away from each other in the sequence that are interacting. And this becomes important when you're interpreting the PAE plot of protein-protein interactions. Then we're talking about things that are away from the diagonal that are coming out as having low error, right? So high confidence, very blue in the plot. The PAE is not a good measure for things like secondary structure, like the PLDDT. So here's, for example, this alpha helix is hard to interpret from the PAE plot alone if it's well predicted. So for that, if that's what you're interested in, then you'd rather look at the PLDDT than the PAE, right? So let's look at this for AlphaFold multimer. So this is the same example. We have our light intermediate chain. This is binding the heavy chain in diene. And so if we want to run a multimeric prediction of this interaction, we can take fragments of these proteins and see if AlphaFold can figure out the interaction between the two. Something important to note here is that if you're using any implementation, either the LMBs or something online, whenever you give AlphaFold more than one chain, it will by default use the model that will predict multimer, right? So we use a multimer model. And there's been a few of these multimer models that have been released with improving accuracy. So V1 was the earlier ones. You often got these weird clashes that would happen. I don't know if people ran those ones and remember. But now with version 2 and version 3, which is the default, it's a lot better. So if you ran predictions in the older versions and you're still interested in, I would run with the newer versions, which are default now. And so yeah, just be aware of that. So this is what a PAE plot looks like. Again, these are probably more informative than the PLDDTs for these protein-protein interactions because we're asking about pairwise, confidence. And these can be a bit difficult to interpret, especially because on the x-axis we have 0 to 600-ish, even though these are two proteins. So it should go from 0 to 300 and from 0 to 300 again for the two proteins. So the way that it's plotted is not that intuitive. But let's just look at one more closely. So we have this one that's the highest ranked PAE plot for this interaction. And then essentially each quadrant is its own PAE plot that I showed earlier, right? So this is the PAE plot of just protein A, right? So this is the confidence of the residues within this chain. And then on the bottom right, we have, again, in isolation, just a PAE plot of protein B. So how confident are we in the interactions within protein B? And if we're interested in the protein-protein interactions, then we need to look at the diagonals, right? So these are the things that are on the top right and bottom left quadrants. And these are asking what is the confidence of the residues in protein A versus protein B and vice versa. And we'll talk about the asymmetry between these two later. But essentially, that's where we want to infer if we're confident in the interactions. If you need help interpreting the PAEs, I find things like Chimera X very helpful. You can just load in the JSON file that has all of this information, and it will open up this thing, which is colored differently, but essentially is the same thing. And you can just drag and outline a part of the PAE, and it will highlight on the structure where you're interpreting, right? So this will help you see exactly, what is this high confidence region? This is a lower confidence region, and it makes sense because it's a loop with the alpha helical bundle on the other side, right? There's another one called PAE Viewer, which is a web app. So this one you can just load up, again, your PDB and your JSON. This one's nice because it shows you this, it highlights the sequence at the same time as well as you're highlighting the PAE plot. You can also load mass spec crosslinks, and it will map them onto the structure and onto the PAE plot. And so you can infer, which crosslinks are match, essentially. I forget the word now, but yeah. Okay, so those are the outputs. We have our 5 structures, we have our MSA output, and we have our quality metrics. Okay, so how do you run your own predictions? The easiest way to get an AlphaFold prediction is to just fetch one that's already been run for you. So we have an AlphaFold database. They've run many millions of proteins now. So if you have, it's even integrated into the UniProt. So if you're looking in the UniProt protein, there's a part where you can just check, an AlphaFold prediction. And sometimes they're really nice. For example, here is just a random one that I pulled up. This is RL3. We have a very nice prediction in the UniProt shown, and it's mapped. It's colored by this PLDDT, which might matter to you, but essentially telling us that this prediction of this globular domain of RL3 is probably right. And you should be aware that, this is put next to experimentally solved structures, and you should just be careful that there might be an experimentally solved structure that's actually more informative than an AlphaFold prediction. You should also be aware that there might be a homologous protein that has an experimentally solved structure that might be more informative than an AlphaFold prediction of your structure, right? So just, these aren't usually displayed in UniProt. You don't know that, the mouse version of RL3 actually has a crystal structure or something like this. So you just kind of have to be aware and do your own digging. What's not useful about this is when you have something that's like an obligate dimer or it doesn't exist in isolation, right? So here's a coiled coil protein, BICDR. And, it's like a long alpha helix here. Of course, we know BICDR is a coiled coil from our, we work on BICDR in the lab. And so you would rather run a multimer prediction of this with 2 copies, but this isn't something that's currently implemented in the UniProt. So somebody might naively look at BICDR and say, oh, it's a long alpha helix, when in fact we know it's a multimer. So usually you want to run your own sequences, right? And so the easiest way to run your own sequence without much effort is to just go onto these online notebooks. So these are Google Colab notebooks, where you can just go on Google servers and put your sequence here and then hit go, and it will use Google's GPUs to run your prediction. This is good for like quick and dirty things, short predictions, small multimers. And these were developed by the Colab team, the AlphaFold team. And they have other, implementations. So they have the AlphaFold with the full multiple sequence alignment. They have this one, which is the AlphaFold with the MM62 that I mentioned is faster. And so that's what we call ColabFold. But they also have other things that are notebook based that are very useful because they're easy to use and don't require much setting up. There's a bit of a limit to this. So, you have to leave the page open, otherwise it times out. So if you have long things, it might not be very useful or might be annoying that it crashes. Also, you have a limit of about 2,000 residues unless you have an account with them and you have their expensive GPUs. And so at the LMB, what's nice is that we can use the LMB's cluster to run, these larger jobs and let it run in the background in the Qt system. And so this is handled by the scientific computing team. I urge you to go to this website and check the guidelines for the scientific computing, how to use the cluster, how to register, fair usage and things like this. It's all outlined really nicely. If you haven't looked or registered, I would, please visit that. But essentially what you need to do is have ColabFold in your path. So this is so that you can run the software. You need to be able to, run it in your terminal. If you're not sure what this is or what this means, you can always email me and I can send you these things, right? But essentially, once you're able to access the ColabFold command, you just need a FASTA file. It has your sequences, one or many sequences, and then you can submit your job from one of the supercomputers to our cluster using a very simple command, ColabFold file, right? And so that will run it in the background. You can come back later and see what the outputs are. Again, it's good to monitor if the things crash so that you're not hogging GPU usage and things like this. So make sure you visit the scientific computing website about that. And so what this will do in the background for you is take your sequences, run it through the MMSeqs to get the multiple sequence alignment, put it through the models, do the predictions, recycle, do all the things in the background, and then spit out the 5 predictions along with the quality metrics and all this stuff. I think it's very important to note here that the way it's currently implemented, the MMSeqs search is handled by a web server. So this is it's handled out of the LMB, right? Sent to web server. And so sometimes, because there's a lot of people running predictions at the LMB, we can be throttled by the web servers. So what we're doing now is trying to figure out how to just do the MMSeqs locally. This is not something you really have to worry about, but just think about if you're waiting in queue to not just submit a lot of jobs that are being throttled. Anyway, we should be able to fix that very soon. Okay, so some of the things that you can play with. So this is getting to your point now, finally. One of the things is you can add as options either to the online notebook or to this, LMB implementation, is change the number of recycles. So remember, the more recycles, the better, up to a certain point. And here's an example of a protein that is being predicted that only really plateaus at like 12 recycles, right? So the previous default of 3 recycles seemed to not be enough for a lot of cases. And so now with the current implementations, it'll continue recycling until it no longer improves, or if it hits 20 first, so whichever comes first. but you can directly change this number if you wanted. There's also the ability to provide a custom template. So this is a PDB file that has a structure or confirmation of a protein that you're trying to predict as a kind of a baseline for what it's going to eventually predict. So it's going to use this information. I find this not as useful. I don't know if anybody here has had success with custom templates, but it's available for you if you want to try it. There's this idea of confirmation and being able to find different confirmations of proteins. I mean, one of the caveats of this is that AlphaFold usually predicts, an open state, and it will fail to predict some closed state of a protein. And there's been many ways to try to extract these different confirmations from the predictions to try to trick it, essentially. And one of the really helpful things to do is, it turns out, is to play with the multiple sequence alignment. If you reduce the number of sequences in the multiple sequence alignment, it essentially introduces uncertainty into the models. And so they can deviate from those minima, so to speak, in those predictions, right? So by reducing the MSA depth with this option, you might be able to pick out those different confirmations. The number of seeds is something I mentioned in response to your question, is for each model, if you give it another seed, so this is another, I mean, don't worry about what it means. It just means that the inputs are going to be slightly different going into the model, and therefore it's going to kind of introduce more variation. And you can have, I think, is there an example here? Yeah, so here they have, I don't know how they go up to like 40 seeds, 30 seeds or something. And in this case, they've also used dropout. So this is another option. Again, don't worry so much about what it means. It's the kind of thing where you just have to try everything, right? And this can also introduce variation in combination with increasing the number of seeds. Another thing you might be able to do, which is, I mentioned in response to your question, was relax the side chains so that they don't look so strange. Sometimes they're super clashed and strange. Even though the geometry is more or less correct, the relative geometries are kind of awful. So if you're fitting into a cryo-EM map, it's useful to just turn on this. There's not that much computational cost necessarily to including this into your pipeline. Exactly. So you can give it a custom MSA. And I should have put this actually here as a way of, you're curating your own MSA is especially useful if you have something like a divergent organisms. Apparently that works really well. And if you want to reduce it, but you want to choose exactly which species or which isoforms, let's say, then you can just use the custom MSA option and provide it with an MSA file. Yeah, so that's a good question. Alrighty. Something that we've implemented recently is AlphaLink 2. So this is an implementation of cross-linking mass spec data into the AlphaFold pipeline. Because if we think about cross-linking mass spec, we're essentially also getting positional information between residues, right? So we should be able to put this in as weights on our multiple sequence alignment to improve the prediction to more closely match what we have from experimental data. And so this was implemented by the Rapp-Silber lab. And here's an example where they are able to get this interaction between these two proteins that more closely matches the crystal structure when they introduce the crosslinks. And so if you want to use this, it's as simple as using collab link sequence, but now you also give it the crosslinks. And in terms of how this should be formatted, you can, with all these things, you can just execute this command on its own, and it should explain everything that you need to give it. Okay. All right, so let's talk about some caveats and some potential issues. One of the first things, or one of the main things that people run into is that they want to predict, these very large things, and there's not, they run out of memory, and there's not enough computational power, and it takes forever. And so just to give you an idea, on the GPUs that we have, you can get, around 3,000 residues total residue counts. And on the notebooks, it's of course less. And for something like what I work on, which is the dining complex, I mean, even the dining heavy chain alone is already beyond these limits, right? So the way that you can approach this is, or you should approach this, is by fragmenting this into different pieces, right? If you're interested in predicting something large like this, yes, it's good to split it into different pieces. Sometimes it's actually helpful to fragment a prediction. For example, here we have BICD2, which is another coiled coil. If I predicted the whole full-length molecule, you have this spaghetti between these two coiled coils. But if you fragment it, you properly get this intermediate coiled coil that we know from other structural evidence that should be in the structure. So sometimes it does improve. I'll show later also fragmenting by fragmenting the protein. Something else that's annoying is a false negative. So these are predictions that should, the interactions that should occur but are not predicted. So here's an example where we have good data on these two proteins interacting, light intermediate chain and the big TR, cold coil. But the PAE, what we're looking at in the PAE is these corners here, right? So we're looking at protein-protein interactions. So they're off-diagonal PAEs, and there you can see they're all red. I mean, you can just look at the model, the atomic model, and you can see that they're not interacting, but it's good to talk about. And for something like this, a false negative, you can get lucky and play around with the way you fragment it, and now you can get something that has a nice PAE. So somehow it's not as distracted by the other residues, I guess, in this prediction. So it's worth playing with the different options to see if you can entice it to properly predict these things. What's I guess more concerning is a false positive. So these are things that AlphaFold predicts, let's say, to interact, but don't actually interact in reality. So here's an example of, again, this light intermediate chain peptide that is predicted to interact with this EB1 protein. You see the PAEs in the corners here are quite high. The PAEs are low, excuse me. So the confidence is quite high. Whereas we have biochemical evidence that these things should not interact. And this kind of thing occurs or can occur when you have a family of proteins where one member of this family interacts with the protein with another protein, but another member of the family does not interact. And because this is a conservation-based algorithm, sometimes you can be fooled by families within proteins within the same family, and therefore getting interactions. And we've seen this a few times with other proteins that should not interact, even though their family members do interact. Yeah, I mean, you're absolutely right. It's possible that the biochemistry is wrong. But what I would say to that is that I think there's multiple examples where even at high concentration, like gel filtration based high concentrations, still not getting interactions, things that you would expect to be useful in a biological context. So I would say this interaction is probably not useful in a biological context based on the evidence that we have. But it's possible that it's worth further investigation. I mean, that's up to you, I guess, depending on your system. What I can say, what is positive here, is that with the newer versions of AlphaFold Multimer, it has become better at not giving these false positives as much. So I like this example too for other reasons, but just to show that the improvements in the Multimer versions, this is the default now, leads to a little bit more skepticism in this interaction, and so it looks a bit weird. If we look at the model, I mean, this doesn't look realistic. And this brings me to my earlier point, or your earlier point, which is the PAE plot is asymmetric, right? So we have on one side, it's kind of confident, and then on the other side, it's not confident. So A versus B and B versus A should be, you would think would be equivalent. And I don't have a satisfying answer as to why this might be the case, but let's just kind of investigate a little bit more closely. So here, if we're asking, what is the error in A50? So let's say A, what's the error in A if we align things at B? And we're not very confident when we align those things. But what is the error in B if we align at A somehow is confident? And it turns out it's because the local conformations of one is less certain than the local conformation of the other, and that somehow leads to this difference. So if this is, if there's a high error in this part, and we align here, then we're probably not going to be correct here. But if there's a high conformational difference in this part, and we align here, we're probably still going to be right here. So I know that's not very satisfying, but essentially I would treat this as a warning flag for the confidence in this prediction. I would be somewhat skeptical. And I mean, with all these things, we have to do further validation biochemically. So here's another example of a false positive. So here what I've done is I've taken a protein and I've fragmented it, but I've kind of cut into a domain and exposed this series of beta sheets that now my other protein has found a way to interact with, right? So you can see here it's kind of stacked on. Again, not necessarily meaning it's false, but probably unrealistic based on the fact that this protein, these beta sheets should not be exposed when the protein is truly folded. So I would be careful with how you fragment things. Even though and the PAE in this case is actually symmetric and confident, right? Something that you should be aware of is a model-to-model variability. I mentioned earlier that the 5 different models somehow are ranked arbitrarily, and so therefore it's good to look at different ones. Here's an example where the highest ranked model has this interaction between this peptide and this cold coil. But if we look at the lowest ranked model, it also has a well-predicted interaction or high confident interaction, but the interaction is actually different, right? So this is probably worth further investigation, whether or not there might be these two parts of the protein might bind. Again, it's hard to tell, but worth looking at because we're not sure which one is true. It turns out that one on the left is true, but it's something that you have to think about. You can also get lucky and get these different conformations of your protein. So this is a cold coil that's kind of folded over itself. And then if we look at the other rank models, some of them have them open, right? So this, especially for a protein like this, where we might know that there's an open and closed conformation of the cold coil, perhaps these are places that one can look to make mutations and follow up on with further experimental techniques. So those are kind of a random list of caveats with these things. All right, so the next thing I wanted to talk about is some applications of structural predictors. One of the main uses, probably a lot of you have used AlphaFold for, is fitting into low resolution maps. So here's an example that Clinton came up with or had, which was this, again, part of this dining complex. We have this dinactin, which is this large, again, multi-subunit complex that has these cold coils that are stretching out, and it's all quite difficult to solve with cryo-EM. So they had this map that was difficult to interpret based on current structures. There weren't crystal structures of these globular domains or anything. And at the time, he ran an AlphaFold prediction of this bit and got this mess. So this is what, again, the older multimer versions would give kind of this messy prediction. And with the newer ones, even, it's hard to kind of fit in these things. And so, again, I would recommend that you fragment these things into smaller bits. They run faster. They're usually more accurate. And so you can get all of these different parts that are much more easily fit into the cryo-EM map, right? And so now they can infer, what are the interactions between this part and this other part of dinactin, make mutations and test if this, inhibitory mechanism is actually, preventing the activity of this protein. Another example is, low local resolution. So in the dining heavy chain, we have a part of it, for example, here that just shows a random place where we have these loops that have, a little bit more difficult to interpret in terms of building a model into it. And so you can, for example, run an AlphaFold prediction of just this little subdomain of dynein, figure out the a first approximation of the side chains, and then stitch that into your model. Now you have something to work with that you can then further, build properly, further refine, I guess. I've also used it for construct design. So this is an attempt at purifying this protein BICT2, which we work with a lot of cold coil proteins, if you can't tell. And so this one, I created a truncation around this 4 helical bundle, which I didn't know was a 4 helical bundle at the time. If you just look at the predictions online, it will say the cold coil ends essentially here. And then so you should be able to just truncate after this cold coil. But if you run the AlphaFold prediction, you can see here I've colored just from N to C that it kind of goes, wraps around and forms this whole 4 helical bundle. And so when I truncated it, essentially I broke this interaction and probably exposed something that made this protein unhappy. So this was insoluble. So then after seeing this prediction, I made a truncation that was further down, and now this protein purified quite well. So this is, I think, an easy way that AlphaFold helps with just biochemistry pipelines. Something that AlphaFold helps with also is just kind of fishing for new interactions. So if you have a biological system and you kind of don't know what is part of a pathway or part of a complex or what's regulating, something you can do is run interaction screens where you have a list of potential interactors, like candidates that come from a database or something. And you can run something like alpha pull down, where you're essentially doing a pull down using AlphaFold. So you're running a bunch of predictions, seeing which ones interact. And then from that, you can hopefully get a list of candidates that you can further investigate with other techniques. So I've written a version of this called AlphaScreen. And so this, what's nice about it is that it just takes uniprot IDs as input. So you can get a list of uniprot IDs for a potential protein using like the biogrid, for example. It just gives you uniprots, and then it will fragment them into little bits with non-overlapping regions, and you can run the predictions on our cluster. And so the kind of output you get is something like this, where let's say we have hundreds of predictions, and we get our top ranked ones that have very nice PAEs, and we can really look into these things and see if we should further study them with your favorite techniques. something is now that becomes very important is how do you interpret a model's quality relative to another model's quality, right? We have 100 predictions. Each one of them has 100 proteins, protein-protein interactions. Each one of them has 5 protein predictions. And we want to rank them all so that we can really only focus on the important bits. And there's like, many, many different ways to interpret the quality of protein-protein interactions that are developed by the predictions. And these are just a few that I've listed here. We have the local interaction score, the protein interaction score. We have DocQ, which is an older one, but also very useful, and a predicted DocQ. So these are either interpreting the atomic models that are predicted, so just looking at the interfaces, or it's interpreting the PAEs and the PLDTs at the interaction interface. And so depending on which implementation you use, you can rank these in different ways. What I would do typically is anyway look at the top parts of this list and not worry about the absolute rank. I wouldn't say that this is necessarily a better interaction than this one, but these are just useful in bringing up the potential candidates from the noise of the sheer number of predictions that you have. And so these are things that you can look into more, but essentially they're all just used for ranking. Something else you can use going through this list is structural homology. So this is if you have a structure and you want to find other structures that are similar to your structure. And these are things that are not necessarily homologous in their sequence, but they're homologous in their structure, right? So these are things like DALI. It's this database. You can just give it your sequence, and it will look at the AlphaFold database. It will look at the PDB, and it will give you a potential list of structurally homologous proteins, which might be useful for your application. There's also FoldSeq, and very recently Progress, which was updated I think this week, and it's much a faster version of these other things. So you can very quickly search through these databases. And so if this is something that interests you, I would urge you to look through these. I'm going to kind of finish off this bit by talking about some of the limitations. Just having them on one slide is useful, and then some advanced use cases. We kind of touched on a lot of these, but the isoform specificity problem is difficult. Again, the isoforms will look similar. It's hard to they're all kind of conserved, right? They're all related to each other. And so getting isoforms to could be predicted differently is very difficult. We talked about oligomerization state, but what I didn't mention is, sometimes you can give AlphaFold a cold coil and you give it 3 copies, and it's like, yeah, sure, I'll make it a cold coil with 3 alpha helices, right? So the question of how to infer the oligomerization state purely based on prediction is still up in the air, I would say. We talked about alternate conformations, the fact that you can reduce the number of sequences in your MSA or give it a custom MSA, and that might help. But there's also these other ways of being a little bit more intelligent about this and taking your whole series of sequences in your multiple sequence alignment and using techniques to cluster them so that you can run different clusters of sequences from your MSA as separate predictions. And now this is apparently a very nice way of, and we've tried it, I think, it's a very nice way of being able to predict different conformations because they sometimes cluster in different homologous proteins. And so there's AF cluster and ACE that can do this. Some limitations are, post-translational modifications are not handled at all, really, by AlphaFold. There's some dedicated models or neural networks that are good for glycosylation. I think there's one for phosphorylation called DeepPhos, but I think they're quite preliminary still. So I think this is still something that is under development. There's also the question of mutations. I mean, we know if we have a point mutation in a protein-protein interaction, it should break it. But because this is a conservation-based thing again, then the conservation of the surrounding residues is actually retained, right? So often what you'll see is you mutate a residue and it still interacts. And that's, of course, because it's still picking up that conservation. So they're not handled quite well, especially in AlphaFold. Apparently, there's good success with RosettaFold. If you're interested in this kind of thing, trying to predict what the effect of a mutation, then you might want to try RosettaFold and have a look at this paper where they address this directly. Something that a lot of people are probably interested in is non-protein interactors. So these are things like ligands and nucleic acids. Of course, a lot of our complexes have these things. AlphaFold will just kind of leave a hole where a small molecule should bind, which is useful for things like AF2 bind or AlphaFill. They can then interpret those holes and fill in with the proper ligand or small molecule. So I haven't tried these myself, but these are worth investigating if you're interested in that. There's RosettaFold 2 and A for nucleic acid. So this is an implementation of RosettaFold that's good for nucleic acids. Apparently performs quite well if you're interested in that. with proteins, I mean. And then most recently, there's this RosettaFold all-atom, which is able to incorporate small molecules, ligands, nucleic acids, proteins, all within the same pipeline. And so I'm trying to implement this now to use to use the LMB's resources to be able to run, these larger things. So they require a lot of memory and such. I would say that this is a promising avenue for all of us, is to be able to just predict all sorts of molecules together. Antibodies are a special case of this because antibodies rapidly evolve, right? That's the way that they work. And because they're rapidly evolved, they don't kind of fit this kind of conservation-based techniques that this algorithm needs. And so it's quite, AlphaFold especially is a quite poor predictor of antibodies. But there's dedicated ones like AB Looper and IgFold if you're interested in this. And apparently RosettaFold now is dedicating to being better at predicting antibodies and even designing antibodies using RF diffusion, if that's something that you want to investigate more. And I mentioned also earlier proteins with low homology, where you can give custom MSAs, or you can use these protein language models that don't require an MSA. But because they have the evolutionary relationships kind of embedded within them, they don't often perform as well as you would expect for something that would not require an MSA, because it's kind of, the MSAs are kind of part of the model, right? So, but it's something that is worth trying if you have something with low homology. There's different ways of thinking about how to cluster. I'm not sure exactly how AF cluster, Mar, do you know how AF cluster clusters? Yeah, so I don't think it's phylogenetic based, but I think it's PCA based, I'm not sure. But I don't know exactly if it's explicit what it's clustering based on. But if you have something explicit like phylogenetic, or if you want to manually curate MSA clusters, then that would be a way to go forward. Yeah. I only have one last little thing I want to talk about, which is just to take you through a few of the papers from the LMB that have used AlphaFold, just as a nice example. So here's just one from the Barford group on the structure of the kinetochore protein NDC80, and you can see this is the NDC80 bit bound to the side of a microtubule. It's a beautiful structure. And in order to help them interpret their map, they could run AlphaFold predictions, fit it into the map. This is a classical use, right? We fit it into the map. We can see which residues may be interacting. Then they can do some other cell experiments to make mutations and verify that these interactions are actually important for this. So this works quite nicely. In Jan's group, they have this AlphaFold-based study where they're trying to generate hypotheses about how cohesin works based on what AlphaFold is predicting, because it's quite complicated and quite a flexible system. And so by generating all these predictions and inferring them, they can come up with these hypotheses that can then be tested later, which I'm sure they're working on. So very nice study, this one. We have from the Hegde group, the structure of the ribosome, and they're trying to figure out how tubulin is autoregulated. And they had these SCAPR and TTC5 proteins bound to the side of the ribosome, and they're trying to, they wanted to know exactly what these interactions are, and to help them interpret the map again, it's a classical use, right? They use AlphaFold, and they can get the residues, and they can see if they mutate those residues that interact with the ribosome, they lose this tubulin autoregulation. So they really helps figure this out. And finally, I'm going to end with one from our lab. So this is, I'm in the Carter lab, so that we work, of course, on dynein, and this is one of the dynein structures previously. We have a LIS1 molecule, which is bound here, which was known to be a dynein regulator, but they had this novel interaction that hadn't been seen before. The map was not very helpful, but they can run an AlphaFold prediction, make a mutation, see that if you mutate this regulator, you no longer get dynein that's running along. And so we've lost the ability to form these complexes robustly. And so also very important. They can do this in cells as well, and see that in cells they lose dynein movement. These are just a few. Of course, there's a lot of the AlphaFold usage at the LMB. I just thought I would run you through some of the classic ones. Okay, I'm going to leave you with this list of resources. There's a listserv where I post updates about implementations and things like this. I also encourage you to kind of discuss predictions, and if you have questions, you could post them on there if you feel like it. And we have these, I think, very useful videos on explaining how to interpret AlphaFold structures, how it works, what they are, things like this. And I'll leave this up, and I'll take any further questions if you have them.